today we are very fortunate to have Dr. Kamila Naxerova as a speaker in our grand rounds. And Dr. Naxerova is an assistant professor in the Center of Systems Biology at Massachusetts General Hospital in Harvard University. She has received Dr. Naxerova received her PhD in human biology and translational medicine from Harvard University and completed her postdoctoral training with Dr. Stefan Elledge. Dr. Naxerova is one of the pioneers in studying somatic evolution in cancer. She has received numerous awards. In particular, she has presented several landmark studies in uncovering the metastatic seeding history through the analysis of phylogenies of clinical samples. Her research had provided molecular evidence that liver metastases of colorectal cancer originate from distinct clones that differ from lymph node metastases. Her lab also discovered that distant metastases experience a more stringent evolutionary bottleneck than lymph node metastases. In addition, more recently, her lab also is expanding the potential of computational and high throughput experimental approaches to understand the history of diseases other than cancer. For example, she also reported increased cell division rates of hematopoietic stem cells in atherosclerosis. Today, she's going to share some of her great findings, and the title of her talk is on the evolutionary history of metastatic cancer. With this, I would like to, hand over this to Dr. Naxerova.
Thank you so much, Ruping, and thank you for having me at your grand rounds. I feel very honored. I understand that a lot of you come from diverse backgrounds, so we may be having some clinicians here, maybe some pathologists who are my favorite kind of clinician because I work with them a whole lot, and then maybe some researchers. So I tried to keep this talk fairly broad, and I'm going to try to talk more about concepts and keep the technical issues to a minimum. And so I hope that this will really enable us to maybe have an interesting discussion at the end where you can ask questions or maybe share some thoughts that you have. I think that I love presenting to clinicians in particular because they have such a unique view on cancer that, I think as a researcher, you don't easily come by. So, any thoughts that you may have at the end of this will be really appreciated. So,
that's great.
Okay, perfect. So as Ruping said, my talk today is on the evolutionary history of metastatic cancer. And so my lab, since it's began its existence a few years ago, has been interested in particular in one topic, and that is metastasis heterogeneity. And as you may know, this is actually something that's really severely understudied. So we don't really know much about how different metastases that exist within one patient are related to each other. And so I want to take just a few minutes to just explain to you why I think that this matters and why I'm really interested in answering some questions in this area, because I think that both for the researcher and the clinicians, there's still some really important things to understand here. So as you've probably heard, and there's a big and kind of thriving research area that has developed around this topic in the last few years, the primary tumor is a genetically really diverse creature. So here in this picture, that's shown by just the different colors in the circle that represent different genetic species or subclones that exist in this primary tumor. And the reason that the primary tumor is so genetically diverse is that it tends to be very old. So we now think that the evolutionary history of a typical primary tumor can start as early as childhood or adolescence. It will evolve out of clonal expansions that exist in the normal histologically functional tissue. And then at some point, it will become a bona fide cancer, but even then it will evolve for maybe a decade or more before it becomes clinically detectable. And so that large time span gives it a lot of time to acquire genetic diversity. So the question that I really think is very important and interesting is, how does this genetic diversity get transferred to metastatic sites? So one possibility that you can imagine is every clone that exists in this primary tumor has equal likelihoods of metastasizing. And over time, what this will lead to is the occurrence of metastases in different parts of the body that pretty much randomly match the abundance of these subclones in the primary tumor. So this would be expected in the case of a random metastasis model. On the other hand, it could also be that there's one particular subclone in this primary tumor that has a much higher likelihood of metastasizing, and that subclone would then be found in a number of different metastases, and they would be genetically really similar to each other, if that was the case. It could also be that metastases are really diverse genetically, but in a way that's unrelated to the primary tumor. And this again would be expected if metastasis occurred really early. So essentially before all this genetic diversity in the primary tumor evolves, if the metastases leave early enough before all of this happened, they would undergo their own parallel evolutionary process and acquire genetic variants that have no relation to what's found in the primary. Finally, it could also be that the rules by which metastasis happens actually differ from organ to organ. So it could be that in some metastatic sites, for example, the locoregional lymph nodes, you find pretty much random metastasis from the primary tumor, as opposed to distant sites where maybe a larger selection acts upon tumor cells, and then they would select for different subclones than you would find in, for example, the locoregional sites. And so I'm going to be talking about this quite a bit today. But I basically just wanted to show you this conceptually, because I think that by studying this metastatic heterogeneity, we can just understand so many things about how the disease evolves. And so here I've listed a few things that I think we can answer if we study metastasis heterogeneity really rigorously. Sorry, just 1 second. Ruping, can you mute yourself? I think that there is some noise.
Sure.
Thank you so much. Okay, thanks. All right, so what can we learn about disease biology if we study metastasis heterogeneity? So as I just said, we can learn about the timing of dissemination, and this is a really important area that Ruping also has made really important contributions to in his work. So that's something that we care deeply about. When do metastases arise? Does this happen right when the primary tumor, right before the primary tumor is diagnosed, or does it happen many years earlier? And by the time we resect the primary tumor, it's all over. So we would really like to know when metastasis mostly happens. We can understand about seeding routes, so we can try to figure out whether one metastasis seeds the other. Maybe this lymph node metastasis seeds this liver metastasis, and then this liver metastasis seeds a lung metastasis. Or maybe it doesn't work like that. Maybe they all come from the primary tumor directly. And so again, metastasis heterogeneity can help us distinguish these different scenarios. And then, importantly in this, I will be talking quite a bit about this today, is we can answer this important question: is metastasis a random process, or do we see selection for one particular genetic species or subclone during the metastatic process? I think that a lot of these questions have clinical relevance. So I think in the end, the rules by which metastasis happens will probably help us understand how to treat it better. In particular, it will help us understand treatment response, which we think will in the end be at least partially related to the genetics of metastases. We can maybe develop better prognostic tools, because there may be different ways in which tumors metastasize, and maybe some are worse than others. And finally, and so this is something that surgeons really tend to care about, maybe we can develop improved guidance for surgical intervention. In some patients with metastatic disease, it may still be useful to resect the primary tumor, and in others, that may not benefit the patient. It could be that different evolutionary mechanisms of metastasis could actually stratify patients between these two scenarios. So that's just a little bit of a more aspirational clinical outlook, but we're hoping that eventually all this work is maybe going to lead to some improvement in these areas. So very briefly, I'll tell you about how I got initially interested in all these topics, and Ruping already touched upon this a little bit, but this is where my kind of historical interest in these topics comes from. So this was during my PhD. I got really curious about this age-old question that every clinician is familiar with. Do lymph node metastases actually give rise to distant metastases or not? And so, as you know, the primary tumor evolves at some primary site. And then for a long time, people have believed, and so this goes all the way back to a surgeon called Halstead, who was doing some really innovative surgeries at the beginning of the 20th century. He believed that the way cancer progresses is that cancer cells first locally invade. They travel through the local lymphatics. They're not strong enough. They're not malignant enough yet to metastasize distantly, but they can take a temporary home in the local regional lymph nodes, and they can start growing there and acquiring further mutations that will then allow them to basically continue on their bad journey and go from the lymph nodes to distant sites. And the reason that Halstead thought this was because there was such a strong correlation between the prognosis of a patient and lymph node metastasis. So invariably, for example, breast cancer patients who were diagnosed with lymph node metastasis tended to do worse than those that did not have lymph node metastasis. And from that, it's reasonable to conclude that maybe there is a causal role for these lymph nodes in metastatic dissemination. But on the other hand, it could, of course, also be, and this is really the hypothesis we need to distinguish here, that the lymph nodes are not really causally involved in the metastatic process, that they're just like a thermometer that measures in a way that is easy for us to detect the metastatic potential of the overall primary tumor. And that if you have a very metastatic primary tumor that sends cells everywhere, it will also send the cells into the lymph nodes where it's relatively easy for a clinician to find them. So either by palpation or at the time of surgery through pathological examination. So these two scenarios are really difficult to disentangle from a clinical perspective. The only way one could do this is to resect the lymph nodes along with the primary and then see if that improves survival down the line for cancer patients. In most cancers, that doesn't appear to be the case. So famous clinical trials in many cancer types, including breast cancer, have shown that the resection of lymph nodes doesn't really seem to be improving survival all that much, with some exception like the sentinel lymph node and melanoma. So at the time, and I think still now, this is a question that we're interested in and that underlies the entire TNM staging system. So we thought we're going to go ahead and take a genetic approach, a phylogenetic approach to trying to answer this question. And so basically, if you can imagine that the primary tumor, is a little family tree of cells that are all related to each other by descent, can we actually, the same way you would do if you want to know who your ancestors are and you send your DNA to 23andMe, can we in the same way figure out what the ancestors of both lymph node and distant metastases are? So are they related to each other by descent? So this liver metastasis, for example, does it appear to be particularly related to the cells that are in a lymph node metastasis? Or in the alternative scenario, can we actually see clearly in the genetics that the ancestors of lymph node metastases and distant metastases are sitting in separate parts of the primary tumor? So in order to do this, what do you need? Well, first of all, you need a great pathology department. You need wonderful clinical collaborators who help you find suitable patients for this kind of analysis. Many of you may know that this is actually really hard to do, because in order to answer this question, we need a lot of different sample types from the same patient. So we need the primary tumor, of course. We need distant metastases, but we also need the lymph node metastases from the same patient. So the tumor draining local regional lymph node metastases that come with that primary tumor and of course, a normal germline control sample. So we searched a lot of patients for these informative pairs, and actually ended up coming up with only 19. That number could be higher. I think it was enough for that study. Nowadays, we'd be a little bit more ambitious probably with these numbers, but they're really very hard to come by. And so at this point, we're looking for samples to study tumor evolution all over the world, just because it's, even at a large institution like MGH, it's really difficult to find the right samples. So, but here in this case, we found 19 patients. And we used an assay that works with somatic mutations in microsatellite repeats in order to study the evolutionary history. And I'll go into how this works in just a moment in a little bit more detail, just so that you know. I think that for clinicians in particular, this could be interesting because you're familiar with this kind of assay for microsatellite instability detection. So it's essentially, like the assay you'd order on a colorectal cancer if you want to find out whether it's microsatellite stable or not, except that it's a much larger number of microsatellites that gets interrogated, and the data is processed in a more quantitative manner in order to understand what mutations in these microsatellite repeats distinguish different parts of the cancer. And so just, very brief background on this. Why do we go out of our way to analyze microsatellites? If you want to study tumor evolution, you need mutations. If you want to find mutations, you can search the whole genome for them and then bankrupt your lab, or you can try to restrict yourself to analyzing a smaller fraction of the genome, but ideally you restrict yourself to a fraction that is enriched for informative mutations. And so if you now look at the rates at which cells acquire mutations in different genomic sequences, you'll see that the unique sequence that you're sequencing, if you're, for example, looking at the exome, actually acquires mutations at a really low rate. So in order to find enough mutations to do phylogenetics, you have to sequence quite a bit. But there are areas of the genome that mutate much faster. And so in particular, there are these mononucleotide guanine repeats. They're just many Gs in a row that mutate really fast. And that's because the polymerase doesn't like them. So DNA polymerase gets really confused when it encounters a stretch of Gs like that. And it tends to drop or add them at a really high rate. So you're basically getting small indels of extra Gs or fewer Gs than you should have. And this happens on the order of 10 to the minus 4 to 10 to the minus 6 per generation per locus. We have now in many in vitro experiments, confirmed this rate. This is really true. It's about 10 to the minus 5 actually, so right in the middle of this range. Depends a little bit on the length of this repeat. But it's very useful to look at them because they're extremely enriched for somatic variants that can help us reconstruct phylogenetic trees. And it's very easy to amplify them and to analyze them. It's the exact same method that your institution will do for microsatellite instability testing. So basically we take fluorescently labeled primers, we amplify these repeats, then the amplicon goes onto a Sanger sequencer, and then we get these characteristics that are distributions where the mode of the distribution corresponds to the true genotype or the true length of this repeat in a cancer sample, and then the distribution around it is this stutter that is accumulated during the amplification, so during the PCR that you're doing with this fluorescent primer. But it's really easy to see even one base pair alterations. So the whole distribution shifts either up or down, depending on whether you have one G more or less. And of course, even bigger mutations are even easier to see, even subclonal variants down to really like 10% variant allele frequency can be detected with this. So it's actually a really, really convenient method. And so we didn't come up with the idea of using these G repeats. There's a really wonderful group at the University of Seattle that pioneered this approach in the mouse. And they had a PNAS paper in 2006 where they showed that in an in vitro evolution experiment where they let cells grow and evolve in culture, you could, based on the genotype of these G repeats, reconstruct the correct phylogenetic tree. So it just is a proof of concept that this is actually, really working. for phylogenetic reconstruction. We have since then, done our own in vitro evolution experiments of this kind. This really works like a charm. It's a really, really efficient way to figure out the lineage of cells that you're interested in. And so you can now imagine that you can do this now for every tumor sample that you might be interested in. Any old FFPE block that's lying around in your department, you can take it, you can extract DNA. It doesn't matter if it is really old and degraded, this will work. This is PCR. And you can essentially, analyze the primary tumor that you're interested in. You're going to be getting back some genotype, and then there's going to be some evolutionary process that gives rise to metastases. And during this process, mutations that have accumulated during the evolution of the primary tumor will expand at a metastatic site, and you will be able to detect them as shifts in this microsatellite distribution with respect to the primary tumor and with respect to the normal germline sample. So it's a really, really efficient method, and we have a multiplex protocol for these primers, so we can amplify a lot of them at one time and get very nice mutational data. Since the first papers that we've published with this methodology, we've really gone all out, both in terms of increasing the density of our sampling and in terms of comparing this methodology to other methods. So here, I know I'm just showing you this is unpublished data, but I think it very nicely shows you the power of this approach. A phylogenetic tree of a patient where we sampled the primary tumor and local regional lymph nodes at the time of surgery, and then also many, many distant metastases at the time of autopsy. And you can see that the tree separates these tumor samples into two main clades, one that consists of almost all of the primary tumor samples and local regional lymph node mets, along with a few lung metastases resected at autopsy. But then most of the samples that were collected that are systemic metastases at the time of death, form their own very tight branch here, indicating that there's a pretty homogeneous clone that contributed to distant metastasis across a variety of organs in this patient. And here you can see the same tree with all the features reproduced. So here the primary tumor and the local regional lymph node metastases, the lung metastases that seem to be matching them just like over here, and then the very homogeneous metastatic clade down here, but this time based on copy number alterations that were determined by shallow whole genome sequencing. You see there's very close resemblance between these two trees. So we really think that this method is very efficient, works very well, and can be done on really pretty much any sample that you lay your hands on in the clinical setting. So just to return briefly to this question now that I had posed just a few minutes ago, what is the phylogenetic relationship between lymph node and distant metastases? Are the lymph nodes actually the culprits in seeding distant metastases? So we collect all the relevant sample types. So here, for example, multiple samples, so multiple spatially separated biopsies from a primary tumor in the ascending colon, a large number of lymph node metastases that were resected along with that primary tumor, and a liver metastasis sampled in 3 different places. And so then you reconstruct the phylogenetic tree based on these microsatellite repeats, and these trees are very easy and intuitive in terms of interpretation. So here are the normal germline samples. They're all very similar to each other. Here's a long branch that represents all the clonal mutations in the primary tumor. And then you can here see, a diverse set of these primary tumor areas that intermingle with a number of lymph node metastases shown in blue as L. And then here are the 3 samples from the liver metastasis in red, along with this one lymph node L3 that is now, of course, very suspicious because it appears to contain the same genetic species as this liver metastasis, indicating that either this lymph node seeded this liver metastasis, or at the very least, they came from the same subclone in the primary tumor. Here's another example of that, which I love in particular. It's very pretty, where you can see that 5 primary tumor samples from a large tumor in the transverse colon all cluster together here in this branch. And then there's another branch that contains a large number of metastases along with this one lymph node here that again is under suspicion for either having seeded them or at the very least belonging to the same evolutionary clade. But in most patients, actually, we didn't see these kinds of patterns where lymph node and distant metastases group together in one evolutionary branch. Most of the time, we could see that pretty clearly the metastases, the distant metastases, were most closely related to a particular primary tumor region, while the lymph nodes were kind of randomly distributed and intermingled with the primary tumor samples, independently of distant metastases. So here, this liver metastasis is closely related to primary tumor area P8, and then the lymph nodes have a distinct evolutionary origin. So across this cohort of 19 patients, we found that really is in a large majority of patients, about 65%, there appears to be no evidence that the lymph nodes played a role in seeding distant metastases. And in only 35% did there appear to be a close connection between lymph node and liver metastases. I will say that this is not really such a super surprising finding in the context of colorectal cancer and liver metastasis, because as you all know, the liver and the gastrointestinal tract have a very close anatomical connection through the portal vein. And of course, it makes sense that a tumor cell that has ended up in the liver probably got there through the portal vein directly by invading hematogenously from the primary tumor. And we have an ongoing study now where we're looking at lung metastasis in colorectal cancer and trying to understand whether lung metastases are more likely to be seeded through the lymph nodes, just because a cancer cell that travels through the lymph nodes would encounter the lung as the first capillary bed that it can get stuck in. So maybe some other time I can tell you about that when we have the definitive results on that. But I'll just switch gears a little bit now and tell you about what I actually think is a more interesting story than the connection between the lymph node and liver metastases, because it points to something more fundamental about metastasis. And so this study really originated after we had published our lymph node paper, that I had just told you about. And we were sitting around and still looking at our phylogenetic trees and noticed something odd. And that was that in a lot of the trees that we had built, we could see that different, so anatomically distinct distant metastases tended to always cluster together on the tree. And lymph node metastases didn't really do that. So here are some examples. Here there's a liver metastasis and a small bowel metastasis, where you see that they have this recent common ancestor. They belong to the same clade here, to the same branch. We call branches like this monophyletic clades, meaning that they are branches on the evolutionary tree that contain nothing but a particular type of sample. So here in this case, a distant metastasis. There's no other samples here, only two distant metastases. So we would call this a monophyletic clade. And lymph node metastases didn't really do that. You see how they kind of branch off on their own. They tend to intermingle with the primary tumor areas like that. Here there is a monophyletic clade of lymph node metastases, but maybe this is not so meaningful because we have 5 lymph node metastases and only 2 of them cluster like this. So we'll get to that in a moment, how surprising is this. But here again, 2 different liver metastases, very closely related to each other. And then same here again, very close evolutionary connection between distinct lesions. And so it really was just striking how differently these metastases types distributed on the phylogenetic tree. And so we began to try to quantify this a little bit. And so it turned out that this wasn't so easy to do. We first began with something like, okay, in what fraction of patients can we find monophyletic clades? So in what fraction of patients do we see that all metastases of a particular kind cluster together in one clade with no other samples? And we found that generally for distant metastases, that was a large fraction. For lymph node metastases, not so much, but maybe that's because we sampled a few more lymph node metastases than distant metastases. And then if you have more of them, the a priori likelihood that they will all be the same, of course, goes down. So we have to account when we when we do this for the number of lesions that are sampled in any given patient. And so, as you can imagine, this number really varies quite a bit, because some people have a lot of lymph node metastases, and then we will, of course, like to look at them all, and some have only 2. And so the same with the primary tumor. Sometimes you have a large primary tumor, and you can sample many, many different areas of it, and sometimes you don't. And so if you want to quantify how much metastases resemble each other with respect to a reference like the primary tumor, you really need a good mathematical framework for doing this, a good computational framework. Otherwise, you may get fooled. And so one of my close friends and collaborators, Hannes Reiter, who is at Stanford and who's also a friend of Ruping's, came to our help with this, and he developed a method which he called the Root Diversity score for calculating just how likely we are to see clustering of metastases of a particular kind on a phylogenetic tree. And so this looks kind of intimidating, but the idea is a really, really simple one. Here, for example, on this tree, we have 3 primary tumor samples that are shown in red. We have 5 lymph node metastases, and we have 2 liver metastases. Now, these 2 liver metastases, they cluster together, and they show, what we call monophyletic clade. They appear to be closely related to each other. What's the likelihood that they will just end up together like that by chance? So in order to calculate this likelihood, you have to count how many other samples do you have. So here, for example, we have 8 other samples. So 3 primary tumors and primary tumor samples, 5 lymph node metastases. And so with 8 other samples on this phylogenetic tree, the likelihood that these 2 guys will cluster together purely by chance is about 6%. So you can just calculate this probability. Of course, if we had more primary tumor samples, let's say we had 8 instead of 3, then it would be even more surprising to see the clustering of these liver metastases, because you have a greater number of other samples that they could, potentially cluster with. And so this root diversity score really tells you, the probability that, an effect like this, where you see a clustering of 2 particular metastases that you're interested in, occurs by chance. So that's really all you need to know. And so now that we have this really nice score that it would account for differences in sampling, we could just go ahead and apply this to all the patients that we had collected with lymph node and distant metastases. And we would just ask, how non-randomly do these lesions cluster on the trees? Another way of saying this is, how different are they from each other with respect to the primary tumor? The higher this root diversity score, the more random they are. So here, all these lymph node metastases, sorry, I should clarify, each dot is a patient. So in this patient, all the lymph node metastases are randomly distributed on the phylogenetic tree. They're intermingling with the primary tumor, no particular connection. That's why the score is so high. And in the case of distant metastases, the closer you get to 0, the more non-random their genetic relationships become. So if the score is really low, it means that the distant metastases all resemble each other, and they don't intermingle with other samples on the tree. So you can see there's a really, really big difference here, indicating much greater genetic homogeneity among different anatomically distinct metastases in distant sites, but not so much in the lymph nodes. So now, of course, you will be asking me, well, Kamila, that's all nice, but distant metastases, they may be metacratously resected. The patient has received treatment in between. Surely you just homogenize them with a particular treatment that killed off all kinds of genetic diversity, except one resistant clone, for example. And that's why you're seeing this severe homogeneity in the distant metastases, but not so much in the typically untreated lymph nodes. So in order to convince ourselves that that's not really the reason for what we were seeing here, we stratified our cohorts by completely by treatment. So here we would compare only untreated lymph node metastases to untreated distant metastases, most of them synchronous. And you can see that the difference actually becomes even more stark, so that the most homogeneous distant metastases are actually the untreated ones. So we don't think that this is a treatment effect. We think that this really has something to do with the fundamental biology by which these metastases are generated. So after we had done this, in all the patients that we had collected for that previous paper, we wanted to know, well, and certainly our reviewers wanted to know, is this actually reproducible, or did you just hit, some sort of odd patient cohort there? So we wanted to see whether, if we just had a completely de novo patient cohort, whether we would see the same thing. So again, we went back to the pathology archives and we looked for patients with primary tumor and matched either lymph node or distant metastases, collected 317 distinct biopsies. I'm just going to show you a few more, I think, informative examples from this new validation cohort, where this effect that I'm talking about is so beautifully visible. So here, for example, patient we call C99, we collected 8 different spatially separate areas of the primary tumor. There was one lymph node that was very similar to the primary tumor as we typically see, and then 2 different liver metastases. Each liver metastasis was sampled in multiple places. So A, B, C just means same lesion sampled in 3 different places, but 2 separate liver metastases. And so what you can see on the phylogenetic tree is that if the multiple samples from the same metastasis are extremely similar to each other, so indicating there's a lot of homogeneity, essentially monoclonal structure within a lesion. But more importantly, these 2 separate liver metastases are closely related to each other. They have this recent common ancestor that is quite distinct from anything that you see in the primary tumor. Here's another example, which is particularly nice because in this patient, we actually had a few adenomas also that were clonally unrelated to a primary tumor that we sampled in 5 different places. So you can see that the adenomas are off on their own evolutionary branch. They have nothing to do with this primary tumor here. And then a number of liver metastases, and then actually 2 other distant metastases that we were able to analyze in this patient. And you can again see how extremely closely related they are to each other, quite distinct from the primary. So for just another example, I'm not going to bore you with all of them, but I'll just say for lymph node metastases, we almost never see that. So the lymph node metastases, they just intermingle with the primary. So this lymph node metastasis comes from this primary tumor area or is closely related to it. And then, this lymph node is somewhere over here where there are more primary tumor areas over there. Some are clustering, but not more than would be expected by chance. So this is what the root diversity score tells us. The way these cluster together, so 2 cluster together, if you out of 5 lymph node metastases with 4 other primary tumor samples present, that's not statistically significant. It doesn't really appear to be a special clustering. Another example here in this case, no lymph node metastases cluster together. They all appear to be coming from separate primary tumor areas, very high root diversity score. So if you now, in this validation cohort, calculate the same root diversity score distribution, you will see the same as we saw in the initial cohort. And that was, of course, very reassuring because it means, we didn't just get lucky or we didn't see an artifact, but really the same effect can be seen here. This is just both cohorts combined. But so in order to understand this a little bit in more depth, and so this is, work that, Ruping will have a lot of appreciation for, I'm sure, because it's a little bit more theoretical. And I'll go over this fairly quickly because it's just a way for us to try to understand a little bit more mechanistically what's going on. We build a stochastic model of tumor evolution where we say, okay, we would like to know what is the relative amount of subclones in the primary tumor that can seed lymph node or distant metastases in order to compare it to our actual experimental data. So we have a number of clones that we're modeling exist in the primary tumor, and then now we just pick some randomly, we pick some of them to be able to seed either lymph node or distant metastases. So for example, in this run of the model, we would pick clone C3 to be able to seed distant metastases, and then it makes a number of liver metastases. And then these 2 clones here, C1 and C6, get selected to be able to seed lymph node metastases. And so then they do that, they send cells to the lymph node. Then in silico, we calculate a phylogenetic tree based on this exact assumption. And we calculate the root diversity scores as we did for our real data. And so we can now vary the number of distant metastatic and lymph node metastatic clones at will, and basically go through any ratio of them that we want. And so what we figured out is that our actual data is most consistent with distant metastasis seeding clones being about 10-fold to 3-fold less frequent than lymph node metastasis seeding clones. So it's just a nice way to somehow translate this fairly abstract data on the root diversity score into an actual ratio. And so I'll get back to this in just a moment to kind of summarize all of this for you, because I know this part is maybe a little bit harder to follow, but I'll just say very briefly, because I think that's important to know. So far, we've been talking about the diversity between different lesions. So if I have 2 lymph nodes and I have 2 different distant metastases, how much do they resemble each other? But the other thing we can be curious about is within one lesion. So within one lymph node, what is the level of genetic diversity there? And so that's actually something that's a little bit difficult to get at with our data, because these polygonine genotypes that we're getting, they're essentially analog data. It's very difficult to decompose that. But we did try to do this here, and I think we succeeded. And this is just showing you what it looks like if we now look at the genetic diversity within one sample. So here, for example, we have a normal tissue genotype for a marker called Nax. I actually don't see this because of the zoom bar, Nax47, I believe. And so this is just one G repeat in the normal tissue of a patient. You can see that there's one mode. So this is a homozygous locus where essentially, the normal tissue has one genotype and the rest is stutter distribution. And so the exact same marker now in a lymph node metastasis, you can see that there's many more little peaks that you can see in the genotype distribution, indicating that there's lots and lots of additional alleles that have appeared in this particular sample. And so here we count 1, 2, 3, 4, 5 different peaks. So the overall, the variance of this distribution is very large. In the distant metastasis, and again measured at the exact same locus, you can see that it's more heterogeneous than in the normal sample. There's big mutation, but it's not as heterogeneous as here in the lymph node. And so we essentially just look at the variance or the variation of these different genotypes that we recover for a particular locus at a particular lesion. And we can use this with some caveats, as in we assume that the copy number status at these loci is similar. Then we can use it to understand subclonal diversity. So here, for example, you can see for a particular patient that in general, the variation of these genotypes appears to be lower in distant metastases than in the lymph node metastasis samples. And so that's a statistically significant difference, indicating that there's just fewer alleles that are present in the distant metastasis. And so we can now do this across all our patients. These are all the patients that I showed you in this cohort, both the discovery and the validation cohort. And you can see that in almost all of them, the allelic diversity is higher in the lymph nodes than in the matched distant metastases. You can do a paired test in order to look at that. And so finally, and I will not really explain in a lot of detail, I'll just say that we did the same thing here by reanalyzing sequencing data from other studies that had sequenced both lymph nodes and distant metastases in primary tumors, and looked at whether we can see that the lymph nodes contain a greater number of variants inherited from the primary tumor in comparison with the distant metastases, and that was the case. So in the interest of time, I will skip this, but if you're interested in this way of looking at intra-lesion heterogeneity, you can take a look at the paper. It's an analysis that shows, as in the previous slide, that within individual lesions, there appears to be more genetic diversity in the lymph node compartment versus the distant metastasis compartment. And then finally, 2 additional pieces of evidence. We always want to make sure that across many different ways of looking at the data, we see the same thing. So here we analyze data from a mouse model of a collaborator who implanted retrovirally tagged cells in the memory fab pad of the mouse. So each cell has its own unique barcode that you can introduce with a virus. And then these cells start metastasizing. You can see how many of the barcodes, how much of the initial diversity that you generated in the primary tumor you can now recover in different metastatic sites. And so this is quantified here. This is the barcode diversity in the primary tumor, and you can see that a lot of it is recovered in the lymph nodes of that mouse. So essentially, many of the cells that are in the primary made it into that lymph node, but cells in the brain, liver, and lung were much more homogeneous. So a smaller fraction of diversity that was present in the primary tumor was able to be transferred to these distant sites. And finally, again, the same result, this time with human single cell sequencing, where we quantified the genetic diversity, so the subclones present in the primary tumor and then in lymph node and liver metastases from a patient with colorectal cancer. And again, seeing the same thing. Primary tumor very diverse, lymph node metastases almost as diverse, and then severe reduction of heterogeneity in the untreated liver metastasis. So again, very consistent. It doesn't really matter if you look in the mouse, if you look with polyGs, if you look with single cell whole genome sequencing, it's always the same story. Distant metastases, more homogeneous. So this is now just a summary of it all. And I think, yeah, I hope that you find this interesting. I always find this absolutely fascinating because it tells us something very profound about the metastatic process. So again, the primary tumor, old creature, as I told you in the beginning, very old creature, contains a lot of genetic diversity, not here indicated in different colors. A lot of the diversity is transferred into the lymph nodes. So different lymph node metastases contain different subclones from the primary, which leads to them being quite distinct from each other. And also within individual lymph nodes, there's a lot of clonal diversity. In the liver, however, very different story. Different anatomically separate liver metastases, even if they sit in different liver segments, which have no vascular connection to each other, tend to be similar to each other, indicating that they're seeded by the same subclone in the primary tumor that is for some reason particularly good at making liver metastases, or they maybe seed each other, but still probably through the bloodstream because the different liver segments have no obvious vascular connection to each other. So really indicating that there's a much stronger selection for a particular subclone in this liver metastatic process. And that selection during lymph node metastasis is much more relaxed. Any old tumor cell can apparently do it in a stage 3 patient. There doesn't appear to be selection for a particular subclone. And so just to leave you with a taste of more to come, I'll just show you 2 more trees before I end. This is some unpublished data, but hopefully published soon, where we just do this now in much more detail across many more patients. So here we again collect primary tumor and lymph node metastases. You can again see how they intermingle with each other. There appears to be no particular lymph node metastatic clone or clade. All these samples here belong to that primary tumor and look original resection. And then here you see samples at autopsy, many different metastases in different sites, extremely similar to each other, with the exception of some lung metastases that don't appear to be part of that same clone, but here mets in different organs, one phylogenetic origin, all very similar to each other. Just a few more examples. If you actually sample the primary tumor really densely, you can actually find sometimes the origin of this bad clone. So here again, many primary tumor samples, some lymph node metastases, all just in their own clade doing nothing. And then all these distant mets, along with some suspicious, actually spatially contiguous areas of the primary tumor, suspicious for containing the clone that then went on to all these distant sites. And just another example of this. We really have a lot of them now, because we're of course very interested. Okay, what is it now about the cells that are in these what we call founder areas in the primary tumor that seed systemic metastases? We really want to get at that, hopefully soon in future work. So I'll end it here because it's 9:50 or 8:50 for you, and I'll just say thank you to my wonderful lab and collaborators. So in particular, Wei-Ting Hang, Alex Orlik and Mati Bloma, who all contributed quite a bit to some of the unpublished data that I showed you today. And of course, our clinical collaborators, who are really just the lifeblood of everything that we do. And yeah, if we have time for questions, I'll go ahead and stop sharing.
Thank you so much, Dr. Naxerova. This is a very wonderful talk, and I really appreciate the thoughtful design that you come up to provide solid evidence regarding the evolutionary history of distant and lymph node metastases.
Thank you.
Are there any questions in the audience?
I have a question from Dr. Scott, excuse me. Go ahead, Scott.
Thanks, Amy, and thanks, Dr. Naxerova. That was a really outstanding seminar. I enjoyed it thoroughly. I guess I have many questions, but I'll ask them one at a time and let my colleagues go right after me. But my first question is whether the increased diversity that you see of clones in lymph nodes, or the increased diversity across lymph nodes, reflects the fact that lymph node metastasis biologically is an easier process than distant metastasis?
I think so. I think that's exactly right. And I have to say, initially I was a little surprised by this because so I'm kind of an immunology naiveling. So I always thought, oh, well, the lymph node surely isn't such a nice place to be because there's all these immune cells that are going to try and kill you. And so that's not so good. But so there was a paper by Sean Morrison's lab that came out in Nature, I think last year or the year before, that showed something very interesting, namely that the lymph is a particularly nice fluid for cancer cells to live in. So it contains a number of compounds, including oleic acid, that protect cancer cells from ferroptosis, and it's like a nice bath that they like to enjoy, and that fortifies them against all kinds of dangers in their metastatic process. So it really does appear that it's just particularly easy for cells to survive in the lymph and then also in the lymph node, and that somehow biologically it's less stressful for them than in the blood circulation. That's right.
Yeah. Okay, Dr. Day has a question.
Oh, yes, Dr. Naxerova, very nice presentation, and I thoroughly enjoyed it. Is it right to assume that all your primary tumors were colorectal carcinoma, or were there tumors from other sites? Yes,
thank you so much. I take this for granted. I don't emphasize this enough. It was all colorectal cancer. So we really focus on colorectal cancer in order to make these comparisons between different metastatic sites fair. And so everything that I showed you was colorectal cancer, yes.
Yeah, I mean, you probably said that, and I may have missed it at the beginning.
I actually probably didn't say it, but so thank you for clarifying that. Okay.
Yeah, I just ask you anyway. I have a reputation for missing things. So the follow-up question to that, it's so even within the colorectal sites, there's just a lot of heterogeneity in terms of tumor types. So the right colon is not exactly the same thing as the left colon, and neither of them is exactly the same thing as rectal tumor. So I'm just wondering how much transferable this data is in terms of what I just said. So do you know the location and the tumor types of this? So that's the first part. And then the second part is the timing of your sampling. So a lot of colorectal carcinoma will have received neoadjuvant therapy. And so is it possible that what you are seeing are those, I think there is enough evidence to believe that lymph node mets occur very, very early. So is it likely that most of these clones were already in the lymph node before the neoadjuvant chemo, and then the subsequent distant metastases were those that were resistant to chemotherapy that subsequently found their way to the liver?
Absolutely. Those are really good questions. And it's good that you asked them together because I think that they are related. So the vast majority of our samples are from the cecum, the ascending transverse, descending sigmoid colon, we have very few rectal cancers. And so rectal cancers will be the ones that are most likely to get neoadjuvant therapy in the form of both chemo and radiation. We have historically not been able to analyze many rectal cancers because in particular after the radiation, a lot of them are just gone more or less. I mean, there's just scar tissue, and it's very difficult to get high purity tumor samples. So we bias against rectal cancers, which I'll say, I don't really have much information on them. The rest though is very homogeneous. So in the collection that I showed you, it's like all in all 40 patients or so. You'll really have both ascending transverse and descending colon. Really, I don't know if it's really equally represented, but a really good mix of all of them. And so in terms of this relationship between lymph node and distant metastases, we think that this really is true across all the different primary tumor locations in the colon, with the exception of the rectum, where again, due to the therapy, we often don't really look at these tumors because they are so heavily treated, and oftentimes it's just hard to find tumor cells. So I hope that answers your question.
Okay, and Dr. Nelson.
Hey, Kamila, this is Andy Nelson. So I'm a molecular pathologist. If you don't mind, I'll start with a relatively easy technical question, and then I have much more interesting questions after that. But just thinking about, how you are measuring these phylogenetic relationships, do you guys have to have any type of a correction within the bioinformatic processes for cancers that are MS unstable versus MS stable? Obviously, there's not a lot in colorectal in respect to hypermutators with a POLE or a POLD1 mutation, but does the background tumor mutation burden of the tumor itself need to be accounted for or corrected for in any way, or does your algorithms in the PCRs to measure these satellite indels just take care of all of that?
Yeah, it's a really good question. We're lucky in that this works really well for both MSI and MSS cancers. So we have two slightly different ways of analyzing these bioinformatically. If we get really high purity tumor samples, we can take the distributions basically as they are, and calculate genetic distances. And then really the only difference between MSS and MSI is that the MSI tumors have a much longer branch connecting the normal germline genotype to the tumor founder. So they have such a much higher burden. I can show you pictures at some point. I mean, the mutations are so much bigger and they're so much more frequent and prevalent. But the nice thing is that these microsatellite repeats, they mutate frequently enough, even in normal cells, so DNA mismatch repair proficient cells, that for phylogenetic purposes, it's almost indistinguishable in terms of accuracy, I think. We now have a new method for analyzing these data that's purity independent. So, again, as a pathologist, this is something that, may be on your mind. Sometimes it's not possible to get exactly the same purity in different samples. And so we've struggled with this over the years, and we did so much work, manually resecting like pure tumor, and every case took forever. So we have a computational method now for accounting for purity. And so that makes the analysis of MSI and MSS tumors even more comparable, because everything gets scaled to a space, so to speak, where the difference to the normal germline doesn't really matter. It's just the evolutionary distance between the tumor samples that matters. So overall, I would say equally good for MSS and MSI, not so good for tumor types where the poly-G repeats just don't mutate. So like in breast cancer, for example, for some reason that I don't really understand, the mutation rate is really low in breast cancers and breast tissue. And so you genotype your little heart out and you just find no mutations at all. So there are some where it doesn't work. So that's the answer.
That was going to be kind of the related technical question, which is, does this work for things that are HRD or, over high-grade serous ovarian cancer is another example where TMB is, sequence mutations don't happen. It's chromosomal abnormalities. And
That's exactly right. That's exactly right.
It seems like something like high C sequencing or other things that capture breakages could be used with similar computational approaches, just measuring a different genomic change. So
yeah, absolutely. And I think that's what you would need. Yeah, exactly. For HRD cancers, these little repeats won't work. You need other
Yeah, you
markers.
can analyze, but the principles I think would be very, very transferable. It'd be very cool and interesting stuff.
Absolutely.
So on that regard then, and I mean, this then gets to the idea of breast cancer. If I understood the overall study design, many of these metastases that you were studying were relatively synchronous with the primary and the primary diagnosis, the primary surgical. Have you had any opportunity to study either A, metastases that are distant that are recurrent, so some time frame of disease, apparent disease-free status before the recurrence, the metastatic recurrence becomes evident, and to study it with your methods?
Yeah, absolutely. We have more and more opportunity to do this now because MGH has this wonderful rapid autopsy program where we're now getting more and more samples from autopsy. And so we've been studying them. In general, the longer the time span between primary tumor resection and metastasis resection, for example, at the time of autopsy, is the bigger the evolutionary distance is. So it's actually kind of fascinating to see that, at the time of autopsy, the metastases that are there, they appear, at least, in phylogenetic terms, almost like a different tumor than what you were seeing in the primary side. So there's a massive evolution going on, I think, both under treatment pressure, but also just over time. And so I think the scales of change are really large. And generally, as I mentioned to you, we generally see a lot of homogeneity. So end-stage disease is pretty homogeneous, more so than in local regional disease, for example, in early stages, or even distant metastases in earlier stage metastatic disease.
Okay, with that, we are going to have to leave, take any questions you have offline. Dr. Naxerova, thank you very much